Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Peter Tarr. I'm BBRF's Editorial Director for Science and Chief Science Writer. And I'm sitting in today for our regular host, Dr. Jeffrey Borenstein, who is BBRF's President and CEO. Jeff had an urgent commitment today and conveys his apologies. Today, Dr. Emma Johnson will present Does cannabis use during pregnancy affect gene regulation in placental tissue? BBRF is the world's largest private funder of mental health research grants, supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. The high quality of the research that we fund is made possible by the BBRF Scientific Council. This group of 192 prominent mental health researchers reviews each grant application and selects the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the foundation to fund creative and impactful basic translational and clinical research that's relevant to the whole spectrum of mental health. One reason that research funded by BBRF has such great impact is because we do not limit our focus to one illness or condition. Since 1987, when we began, the foundation has awarded $462 million to fund more than 6,700 research grants around the world and across a broad spectrum of brain illnesses. And these include addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, PTSD, schizophrenia, as well as suicide prevention. Today, I am absolutely delighted to introduce Dr. Emma Johnson. Dr. Johnson is an assistant professor in the Department of Psychiatry at Washington University School of Medicine in St. Louis. She received a BBRF Young Investigator Grant in 2020. Our webinar will begin with Dr. Johnson's presentation, which will then be followed by a Q&A. To submit your questions, please use the questions tab on the control panel on your screen. And please feel free to submit your questions at any time during the presentation. Following the presentation, I will ask as many questions as possible in the time that we still have left. And so now I am very pleased to introduce Dr. Johnson.
Thank you so much for that introduction, Peter. I think you can all see my screen.
Yes.
So I'm excited. Oh, thank you, Nico. Excited to talk to you all today about prenatal cannabis use and its effects on DNA methylation in placental tissue. I don't have any disclosures or conflicts of interest to report besides current funding support from NIDA and the Evolution Foundation and the American Foundation for Aging Research. Since this is a Meet the Scientist webinar, I just wanted to give kind of a brief introduction of the work that I do in my lab. So we study the genetic and environmental factors that contribute to human behavior and psychiatric disorders, with a focus on substance use and substance use disorders in particular. And we do this by leveraging genomic and multi-omics data at a large scale, applying statistical and computational methods with the ultimate goal of gaining insight into human behavior and health. And to date, my work has primarily focused on genomic studies of substance use and substance use disorders. So I'm going to start with a little introduction there. So when we study substance use behaviors and substance use disorders, these are what we call complex and polygenic traits. What do I mean by that? By complex, I mean that substance use behaviors and substance use disorders are influenced by both our DNA as well as the environment around us. By polygenic, I mean that there is not any one gene that causes a substance use disorder, but rather these behaviors and psychiatric conditions are influenced by many genes and many types of genetic variants all across our genome. So the way that we typically study a complex polygenic trait, or at least the genetic piece of these complex traits, is using something called a genome-wide association study, or a GWAS. And you may already be familiar with what a GWAS is, but I'll give a simple overview of what these studies mean. So in a GWAS, we are typically collecting cases and controls from a population. For example, we might be studying something like alcohol use disorder. And once we've recruited our cases and our controls, we compare their genomes and we look at tiny single nucleotide variations across their genome to see if we can identify genetic variations or these small differences in our DNA sequence that are associated with whatever trait or disorder that we're studying. One way to visualize the results of a GWAS is using something called a Manhattan plot, and that's what I'm showing here on the right. So just to orient you to this type of figure, because I'll show more of these later today, each dot on this figure represents a single genetic variant in our genome. And you can see that these are laid out along the x-axis by their physical position along the genome by chromosomes. So we see here, these are laid out from chromosome 1 to chromosome 22. On the y-axis, we have the -log10 p-value, and that just represents how strongly each genetic variant is associated with, in this case, alcohol dependence, which is what we were studying in this study. So the genetic variants, or the dots on this figure that are higher along that y-axis, are the ones that are most strongly associated with, in this case, alcohol dependence. And much of my work has focused on trying to understand genetic factors that influence liability for cannabis use disorder. So I'm showing here a Manhattan plot from a study that we published in 2020, where we collected cases with cannabis use disorder and controls without cannabis use disorder. And we identified two genomic loci, or regions along the genome, that were associated with greater risk of developing cannabis use disorder. This was more recently updated in a much larger GWAS meta-analysis. As you can see here, we identified far more regions of the genome that were associated with risk for cannabis use disorder, and this was a study led by Dan Levy and colleagues. So we've seen that genetic factors can influence our likelihood to engage in substance use behaviors and our risk for substance use disorders. But can our substance use affect our genome? It's not going to affect the sequence of our DNA, but can it affect how genes are expressed, for example? And so this is where we come to epigenetics. While our DNA sequence remains static throughout our lifetime, our epigenome is actually really dynamic, and that can change. So what I'm showing here is what I think is kind of a nice visualization of how our DNA is actually structured. So I think we often tend to think, or at least I tend to think of DNA as just this kind of linear sequence of letters, right? But it's really this 3D structure that is very dynamic. It has these modifications that I'll talk a little bit about. And so one type of epigenetic modification is what we call histone modifications. So histone acetylation and methylation can cause increases or decreases in gene expression by changing how tightly wound the DNA is around what we call these histones that form nucleosomes. And so if that DNA is more tightly wound and that chromatin is bunched together tighter, then those genes are not going to be able to be expressed as much. Whereas if that DNA is wound a little bit looser, those histones are not as tightly bound, then the genes will be expressed in that region of DNA. Another type of epigenetic modification is what we call DNA methylation. And this is where methyl groups are added to certain regions of our DNA, and this represses gene expression at those sites. And certain environmental exposures, including substance use and cannabis use, can induce these changes in our DNA methylation. And so that's what I'm going to be talking about today. We're going to be focusing on DNA methylation as an epigenetic change following prenatal cannabis use. So there have been what we call epigenome-wide association studies, or an EWAS, of cannabis use. I'm showing here a figure from a study from Vong et al. That was published in 2024. And this is, you'll notice, similar to the Manhattan plot from a GWAS, but here we're asking a slightly different question. So these authors were interested in examining whether cannabis use potentially influences gene expression through changes in DNA methylation that are associated with that cannabis use. And so in this case, the authors found 4 significant sites along the genome that showed significant changes in or variations in DNA methylation that were associated with cannabis use. But I want to take a step back for a minute and just talk about changes in cannabis use, especially in the United States. That will be my focus for today. So as cannabis legalization has expanded throughout the United States, cannabis use has also continued to rise. So I'm showing here a figure on the left, and this is data from Monitoring the Future. And we can see increases in cannabis use for both the age 19 to 30 age group, as well as the older age group, ages 35 to 50, whether we're looking at cannabis use within the past 12 months or even daily use. We are seeing increases, especially in recent years. People report using cannabis to help treat their anxiety, if medications aren't working. They also use cannabis to help with pain, sleeping better, as well as other issues. And many people who use cannabis will not experience adverse effects, but some people will, and this is important. So potential issues involved with cannabis use include symptoms of cannabis use disorder. Cannabis use can even induce acute transient psychosis symptoms. And there can also be more serious conditions like cannabinoid hyperemesis syndrome, which tends to be associated with long-term heavy cannabis use. So we're seeing these increases in cannabis use among the general population, but interestingly, we're also seeing increases specifically in pregnant populations with prenatal cannabis use. So I'm showing here a figure from a paper by Arpana Agrawal and colleagues at WashU that was published in 2019. And in this figure, you'll notice we're generally seeing decreases across the past decade or so in alcohol use and tobacco use during pregnancy, but we're seeing a very slight uptick in cannabis use. And 34 to 60% of individuals who use marijuana in their life keep using it during pregnancy. Many of these individuals believe that cannabis use is relatively safe during pregnancy. They haven't been told anything different. But this is a little bit concerning because the cannabis being used today is not the same as cannabis being used, a decade or so ago. The potency of cannabis has at least tripled, if not more. And that's shown in this figure here. So what we're looking at here is the amount or the percent of THC in cannabis samples, and that's shown in this red line in this figure. And we can see that the percent of CBD in cannabis has remained relatively the same, but the percent of THC, which is the main psychoactive component of cannabis, has shown a sharp increase, right, from around 4% in 1995 to over 16% in 2022. So cannabis is changing, it's becoming more potent. This is a figure from another study that came out in 2019 from Nora Volkow and colleagues, also looking at increases in cannabis use during pregnancy. So again, we're seeing, if you look at the darker bars in those figures, In this figure, that represents cannabis use in the years 2002 to 2003. Compare that to the lighter bars showing the prevalence of cannabis use in the years 2016 to 2017. We're seeing significant increases across all populations, so non-pregnant women and pregnant women. And interestingly, we're seeing a significant increase in the first trimester, but we're also seeing what I would call suggestive increases in the second and third trimesters as well. And here's kind of an interesting excerpt from a New York Times piece that came out in 2017. This woman is talking about how her OBGYN was concerned about whether she smoked cigarettes or used alcohol while she was pregnant, but they weren't really concerned about her marijuana use. She says since it hasn't been studied that much, they couldn't advise her one way or another. So we're seeing these increases in prenatal cannabis use during pregnancy, but it's unclear to the extent to which that will influence birth outcomes, child behaviors, both short and long term. So is there evidence for an effect of prenatal cannabis use? When we look at behavior, generally the literature has had somewhat inconsistent findings, I would say. Relatively small samples have been used to study this, so we don't really have a ton of data yet from larger samples, although I will present some more data in a bit. And it's also difficult to separate the effects of prenatal cannabis use from potential confounders. So for example, other forms of substance use that might also have an effect, as well as differences in things like socioeconomic status or other potential confounders that can make it difficult to kind of separate out the true effect of prenatal cannabis use from these other potential factors. For birth outcomes, I think there's a little bit more data here. So I'm showing a figure from a meta-analysis looking at the effect of prenatal cannabis use on low birth weight in this figure. And we see that for the individual studies, these have relatively small effect sizes and relatively large standard errors. But when we look at all of that data meta-analyzed together, represented by that diamond at the bottom of the figure, we do see a significant increase in risk for low birth weight with prenatal cannabis use. So there is some evidence that has an effect on low birth weight. Haven't seen as much consistent evidence for child behavior. But I want to draw your attention to a study that was led by Sarah Paul here at WashU. She's a grad student in Ryan Bogdan's lab who actually just successfully defended her dissertation. And she led this study in the Adolescent Brain Cognitive Development Study, which is a large longitudinal study of around 10,000 kids across the United States. And what Sarah found was that prenatal cannabis exposure was associated with increased risk of externalizing problems in these children. And this was at baseline. So these children were around age 9 to 10 at this stage. And she found that prenatal cannabis exposure was associated with greater externalizing problems. So this includes things like aggression, impulsivity, potentially substance use down the road. And interestingly, she found that this difference was significant both for children who were exposed after their mother knew about the pregnancy, so post knowledge of the pregnancy, as well as children who were only exposed pre-knowledge or before the mother knew about this pregnancy. Similarly, she found that attention problems were increased in these children who were exposed to prenatal cannabis. So we are seeing some evidence for effects of prenatal cannabis on psychopathology, at least in this sample. It's a relatively large sample. Sarah also found that these effects remained when she controlled for potential confounders, including, for example, genetic risk that might contribute to these issues. And here I'm presenting a follow-up paper led by David Berenger and colleagues at WashU. And David looked again at a similar question: is prenatal cannabis use associated with psychopathology in the children of the ABCD study? But David looked at this at the, I believe, 2-year follow-up. So now the children are ages 10 to 11, and we still see these effects of prenatal cannabis on things like stress problems, obsessive-compulsive, ADHD issues, conduct problems. And he also found that for sluggish cognitive tempo and psychotic-like experiences, these associations were significant, again, when the children were exposed only before the mother knew about the pregnancy, as well as after knowledge of the pregnancy. So we are seeing more evidence collectively that points to potential associations between prenatal cannabis exposure and psychopathology in children. But we really need more research on why this is happening, and to what extent is the in utero environment potentially responsible for some of this. It might be a mediating factor in this relationship. And this is, I think, an interesting, complicated area of study, because THC can pass directly from the mother to the developing fetus, but it can also bind to endocannabinoid receptors in the placenta and potentially alter placental function, structure, and this can then affect things like fetal growth, which might be leading to these associations that we're seeing between prenatal cannabis exposure and risk for low birth weight, for example. I think another potentially interesting path is through the brain. So this is another paper led by David Berenger that came out last year. And again, using the ABCD study, David was able to connect prenatal cannabis exposure to psychopathology in children, but now he's looking at what extent is this mediated through brain correlates. So he found that differences in gray matter and white matter were mediating some of these relationships between prenatal cannabis exposure and things like attention problems and ADHD symptoms. So this could reflect another indirect path from that in utero environment through the brain to these child behavioral outcomes. I also wanted to discuss this interesting study from Rampala and colleagues that came out in 2021. And they looked at perinatal cannabis use and offspring behavior, as well as gene expression in placental tissue. So in particular, for their placental analyses, this was a sample of 30 maternal cannabis users, and they considered self-report of pre or postnatal cannabis use to be broadly maternal cannabis use, and 100 controls. And what they found was that maternal cannabis use was associated with increased anxiety, aggression, and hyperactivity in children at around ages 3 to 6 years old. Interestingly, when they looked at gene expression in their placental tissue, they found reduced gene expression in immune-related gene networks, and this was then correlated with those anxiety symptoms in the children. So this is highlighting a potential role for changes in gene expression and gene regulation in the placenta, potentially through immune-related processes, and then that being a mediator of these later child behavioral outcomes. But we really need more data on prenatal cannabis use, how that potentially leads to changes in the placenta, and then how that connects to short and long-term effects on child behaviors. And so I'm excited today to talk about data from a study called the Cannabis Use During Early Life and Development, or the CUDDEL study. The PIs of the study are Arpana Agrawal and Cynthia Rogers at WashU. And the CUDDEL study was designed to assess the effects of prenatal cannabis use on baby's brain and behavior. So in the CUDDEL study, we recruited pregnant people with a lifetime history of cannabis use. So I think that's a really unique aspect of this study. These are all people that have used cannabis during their lifetime. And then we're separating out people who continue to use cannabis during their pregnancy and comparing them to people who stop using cannabis during their pregnancy. And you can see in this figure that we're collecting data on self-report substance use as well as urine drug screens at different time points throughout the study. We're also collecting data on pregnancy and delivery complications, as well as psychosocial assessments of the mothers. And then in the babies, we're collecting MRI data, neurobehavioral data, data on temperament, cognitive development, things like that. So here is just some basic demographic information about this study, and then some information about substance use in this sample. So this sample is primarily of participants who self-report their race as Black. I would say this is a fairly under-resourced sample. Again, as I mentioned, all of these folks have used cannabis before in their life. You'll see that 80% of the cases, around 60% of the controls, used cannabis in the 2 years prior to their pregnancy. And many of these individuals had used cannabis at least 100 times in their life. In terms of how cannabis is used in the CUDDEL study, about 15% of case participants report that their typical use is a joint, whereas around 50% are reporting blunt smoking, and 60% report vaping, edibles, or other forms of use. In terms of why participants are using cannabis during pregnancy, most of them report that they're using this for helping with nausea or appetite. Many of them report using cannabis to help with sleep difficulties, anxiety or stress management, as well as dealing with pain. And preliminary data from CUDDEL suggests that we are seeing a significant effect of prenatal cannabis exposure on birth weight. So we see that individuals with prenatal cannabis exposure tend to have lower birth weight than those without prenatal cannabis exposure. So that's fairly consistent with the literature. And then interestingly, we're also seeing a significant effect of prenatal cannabis exposure on externalizing behaviors. So this is very similar to what Sarah Paul saw in her study. Obviously, we're looking at a much younger sample here, right? So these are infants and toddlers compared to the ABCD study where the children are 9 to 10 years old. But it's really interesting that we're seeing these effects of prenatal cannabis use on externalizing behaviors at this early age. Today, I'm really excited to talk about the epigenetic data that we were able to collect in this CUDDEL study. So this is all thanks to a 2020 Young Investigator Grant from the Brain Behavior Research Foundation that I received to collect and store the placental tissue from CUDDEL participants and analyze DNA methylation in these samples. So we've collected methylation data on 192 samples. 121 of those have prenatal cannabis use and 71 do not have any prenatal cannabis use. And I also need to thank my co-mentors on this grant, Ryan Bogdan here at WashU and Danny Fallon at Emory, for their support with this project. Our main questions in this project are, is prenatal cannabis use associated with DNA methylation in placental tissue? And do these epigenetic variations that we see potentially mediate associations between prenatal cannabis use and later child behavior? So our DNA methylation data was from the Illumina EPIC2 array, and we performed pretty standard quality control procedures. For our actual epigenome-wide association study, or that EWAS that I'm going to be presenting today, we're essentially testing for an association of variation in methylation at each site in our array with prenatal cannabis use. In these analyses, we controlled for cotinine. So this is a nicotine metabolite. So many of these participants, might be exposed to secondhand smoke, for example, or this might be because of the mode of cannabis use. But regardless, we did have some urine drug screens that were positive for cotinine, and so we're controlling for that in this analysis to try and separate out these effects of prenatal cannabis use from anything that might be, potentially related to nicotine instead. We're also controlling for age and the sample plate. And then I'll also present some follow-up analyses where instead of looking at individual sites along the genome, we're actually aggregating those sites together if they're close together and looking at what's called a differentially methylated region, or a DMR. And then I'll also present some gene enrichment analyses that I think are quite interesting. So within this subset of participants where we have placental tissue that I'll present today, in terms of why they're using marijuana, the patterns within this subset are very similar to what we see within the larger CUDDEL sample. Again, most people are using this cannabis to help with their nausea or appetite. Many others report using this to help manage their anxiety and stress, to help sleep better, or to help manage their pain. So here is our EWAS of prenatal cannabis use. Again, our sample size here is 192 participants. 121 of those had prenatal cannabis use and 71 did not. And again, just as a reminder, we are controlling for cotinine exposure here to try and really get at the effects of cannabis use rather than nicotine exposure. As we might expect with this sample size, we didn't see any individual sites reach genome-wide significance, but we do have some that are suggestive, and I want to just take a look at two of these sites. These aren't particularly special sites, but I want to show you kind of what the data looks like underlying that Manhattan plot. So in these two figures for these two of our top sites, I'm showing differences in methylation within the group of no prenatal cannabis use compared to people who did have prenatal cannabis use. And you can see that there's a lot of variation within these samples, right? But for both of these sites, we do see that on average, for the group with prenatal cannabis use, that methylation is a little bit higher than in the group without prenatal cannabis use on average. There is a difference in terms of how much variation there is in terms of methylation at each of these sites. So for example, the site on the left, that variation is a little bit lower. We have a little bit more variation on the right. But again, on average, at least at these two sites, we are seeing more methylation in general in the group with prenatal cannabis use compared to those without. I mentioned earlier that we also ran an analysis where here we're not really looking at individual sites, but we're sort of aggregating the sites that are close together on the genome and trying to see if there are any regions of the genome that show significantly different methylation. And we did find some regions with significantly different methylation between prenatal cannabis use and those without. And I've highlighted those here. And I took these regions and the sites within these regions and used something called the EWAS Atlas, which is this really nice database of published epigenome-wide association studies. And so you can put your sites or your regions of the genome into that atlas and see, have these been previously associated in other EWAS of different traits? And what we found was really interesting. So some of the sites that we identified in our study have been previously associated with inflammatory related diseases like rheumatoid arthritis, airway inflammation, and asthma. We also see associations, these sites that have been previously associated with maternal alcohol consumption, preterm birth, which again is very consistent with the literature. We see associations with cancer, autism spectrum disorder, BMI, as well as smoking, cognitive function, air pollution, and childhood stress. And I thought this was really interesting because I didn't mention this earlier when I was talking about Sarah's study in ABCD, but she also found associations between prenatal cannabis exposure and BMI and cognitive function. And so it's really cool that we're seeing some of those things pop up as well when we look at this very different sample. So this is what's called a volcano plot. Some of you may be familiar with this, but it's just a different way to visualize our results. So again, here on the y-axis, we have the negative log 10 p-value. So the dots that are higher on this plot represent those that are more strongly associated with prenatal cannabis exposure. And then on the x-axis, we have the effect size. So we can look at sites where more methylation is associated with prenatal cannabis exposure and compare that to sites where perhaps lower methylation is associated with prenatal cannabis exposure. And what we did next was, we didn't identify any genome-wide significant individual sites, but I took the ones that were nominally significant. So in this case, those that had a p-value less than 0.001, and those are shown on the top third of this plot. And I took those and performed an enrichment analysis to see if the genes at these sites were associated with any particular biological pathways, cell or tissue types, etc. And again, what we found was, I think, very interesting in light of previous literature. So we did see that genes were enriched in particular cell types and tissues. So for example, in immune-related cells in the placenta, as well as excitatory neurons and the cortical nucleus of the amygdala, which has been associated with emotional learning. We also saw that they were enriched for signaling pathways that are important for cellular processes. We saw associations with regulation of neuroblast proliferation, as well as the regulation of different cell cycle processes and the presynaptic active zone. And then finally, we also saw an enrichment for respiratory system diseases. So I think a lot of this is pretty consistent with some of the literature. We are seeing these associations with inflammation and potentially immune-related processes. We're also seeing that these regions were previously implicated in studies of cognition and preterm birth. And I think the fact that we're seeing some of this enrichment for signaling pathways, certain brain cell types and tissues, as well as immune-related cell types in the placenta, these may be pointing to mechanisms by which this prenatal cannabis use could potentially influence child behavioral outcomes. So what's next for this study? We have a few questions that I'm really interested in trying to answer. The first thing I want to look at is, do our differentially methylated sites overlap with loci that have been identified in GWAS of cannabis use? We're also, of course, interested in taking this forward and looking at whether these epigenetic signals that we're seeing, are they potentially mediating associations between prenatal cannabis use and child behavioral outcomes, for example, those associations with externalizing behaviors that we're seeing in the CUDDEL sample. And then finally, it would be great if we could potentially replicate this in another sample, potentially be able to meta-analyze in the future so we can increase our sample size and our power to detect these associations. So for example, I'm showing here the logo from the Healthy Brain and Child Development Study, or the HBCD study, where we have collected placental tissues. So the ultimate goal there would be to look at both epigenetics as well as transcriptomics in that placental tissue. The HBCD study is not focused specifically on cannabis use, but I think that would actually be really nice because we could potentially look at, are there differences in terms of these epigenetic signals for cannabis use versus other substances like opioids or alcohol. There's also consortia like the PACE Consortium, Pregnancy and Childhood Epigenetics. I'm not aware of any studies that have specifically looked at prenatal cannabis use epigenetics in the placental tissue. But we could potentially join forces with a consortium like that to try and replicate, or again, look at differences between different substances and how those influence the epigenome. With that, I would just like to thank BBRF so much for their support for this project, as well as my collaborators, my co-mentors Ryan Bogdan and Danny Fallon, as well as collaborators here at WashU, and the CUDDEL PIs, as well as the CUDDEL participants and staff, without whom none of this work would be possible. So thank you.
Thank you so much, Dr. Johnson. We have a number of questions that came up while you were speaking, and I'm going to try to relate some of them to you, and I hope we have a chance to talk about a few of them. Just for purposes of clarification, if I could just start off by going back to something you said right at the beginning of your presentation. You're looking at placental tissue. First of all, how do you get this placental tissue? When do you access it?
Right, that's a great question. So I personally was not involved in collecting this placental tissue, but we have wonderful staff here at WashU who do that, as far as I know, pretty much immediately after birth, as soon as that is available. And they are very skilled in this, so they know how to collect the placental tissue. They store that tissue. I can't remember off the top of my head exactly which parts of the placenta we're getting, but it is collecting from several different sections of the placenta.
I understand, and thanks for that clarification. So let me ask you, forgive me for just going back over this, but it's important. The way, whether it has to do with cannabis exposure or any substance for that matter, nicotine, whatever, how, what is the mechanism of transfer between the placenta and the fetus? Is it through the bloodstream? Is it through blood vessels? How does it happen?
Yeah, that's another great question. Maybe I can go back to that slide actually. Sorry, there are many slides here. Right, so I think, well, I guess this figure doesn't quite get at this, but that's a great question. So THC can pass directly to the fetus through the mother's bloodstream, right? So that is one way that, a prenatal cannabis exposure in THC is potentially influencing the fetus as it's developing. But as I mentioned, it can also, THC can also bind to endocannabinoid receptors in the placenta that can potentially alter like the vascular structure of the placenta. It can alter the placental function, things like that. And so in that way, there's kind of this indirect effect as well on fetal growth. Even though THC can pass directly from the mother through the bloodstream to the fetus, there can also be these changes to the placenta itself, and in that way potentially affect fetal development.
Very interesting. And I'm really glad you anticipated my next question, which has to do with those endocannabinoid receptors in the placenta. One thing that's sort of implied in your presentation, but not made explicit, but we should say it for the point, just so that people who don't know this, we have a natural endocannabinoid system in the body, a naturally occurring endocannabinoid system with cannabinoid receptors that has nothing to do with taking in THC that is plant-based, right? It's built in. We are born with this, the endocannabinoid system.
Absolutely, yes.
And so, yes, amongst many other places in the body, including in the brain, there are endocannabinoid receptors called CB1 and CB2 in placental tissue itself, just natural, which is fascinating. And they're there because the endocannabinoid system helps regulate various things like stress, right? I mean, why do we have an endocannabinoid system in the first place? Apart from taking in extra, endocannabinoids via external sources, yes?
Yeah, I mean, that's exactly right. It is fascinating that, the placenta itself has all of these CB1, CB2 receptors, and they are important for normal placental function, right? So it's not just, right, it's not just, people who are using cannabis during pregnancy. We all have this endocannabinoid system, and it's important for lots of different functions.
Right, and as I understand it, what the problem that can develop, although it only affects a relatively small percentage of users, but people who develop uh, cannabis use disorder (CUD) are people whose intake of cannabis actually sort of overwhelms and dysregulates this naturally occurring cannabinoid system in the body, right?
Yes, yeah, exactly.
And so now, we sort of begin with all of these amazing findings that you've made in your research regarding the relationship between these changes in methylation within placental tissue, for example, and possible behavioral effects in children after they're born. How do you follow? Do you follow the children after birth? And if so, for how long and how do you follow them? What do you measure in them?
Yeah, that's a great question. I think that's something we're trying to do in CUDDEL, right? So the original CUDDEL study, we're taking data at 6 months, 12 months, 18 months after birth, but we're hoping to continue to track these children, through toddlerhood and their early years. And then I think that's something, that the HBCD study is also kind of trying to really capture is, we have the ABCD study, which I think has been phenomenal for, looking at how is this prenatal cannabis exposure influencing these children, in their preteen years when they're 9, 10, up to, 16 years old now. But, how early are we seeing those differences in childhood behavior? And so, again, I'm really excited to look more at this in CUDDEL, where we do have all this data, not only on the infant's behavior and their cognitive function, but also neuroimaging data, which I am not an expert in, but there are people in CUDDEL who are experts in looking at the brain data. And so I think that will be really interesting to see. Are we seeing, subtle differences in how this is affecting these babies' brains, and can we link that to their behavior and cognitive function?
And once again, you've set it up perfectly. My next question would be what has to do with the difference between association and causation. When somebody makes a statement like prenatal cannabis use, as seen in this data, is associated with X, Y, and Z, that's an association. That means people whose, let's say, children whose mothers took in cannabis during pregnancy, perhaps even during the second or third trimester, these children later went on to be measured in various behavioral parameters, and they were different, en masse. They were different from children whose mothers did not take in cannabis. So that's sort of an association, but it doesn't describe the mechanism by which this might have happened, right? Just to establish the difference between, association studies and then ones eventually where you get behind the association and try to figure out the cause.
Yeah, exactly. That is an important thing to note, that, a lot of what I'm talking about here is an association or a correlation, not necessarily causation. Although I will say, I think it's kind of baby steps here, right? For example, when we're looking at the ABCD study, well, I guess it is a longitudinal study, but we can look at cross-sectional studies, right, where we only have data on one time point from folks. And so, all we can use is maybe a retrospective self-report, was your mother using cannabis when she was pregnant, and see whether that's correlated with, with behavior or, whatever we want to look at that particular time point. And then I think, being able to look longitudinally or prospectively we have this sample and we have, carefully tracked how much cannabis use the mothers are using during their pregnancy. At each trimester, we have data on that. We're trying to control for as many things as we can control for, including cotinine exposure and, potentially exposure to other substances, things like that. So you're right, it is an association. It's, we can't draw causation, but I think we are getting closer, and I think these longitudinal studies are kind of a step further in trying to get at the extent to which this might be a causal relationship. And then, of course, we want to dig into the mechanisms. Why are we seeing these associations?
Right, and just based on what you know so far, and we're not going to hold you to this because this is what science is about discovering these answers. But I mean, just based on what you know, since you study this, what do you think some of the mechanisms might potentially be through which prenatal cannabis use can affect the actual behavior of a child years down the road?
Oh, yeah, that's a big question. I mean, I think we have some clues that are intriguing. I think some of the associations we're seeing with, inflammation-related phenotypes are quite interesting. We were talking earlier about the endocannabinoid system, and we know that, one of its functions is kind of related to, the immune system and immune system regulation. And so I think looking more into immune functions and some of these inflammatory phenotypes might be potentially really interesting, and maybe that's one potential mechanism. Again, I also think David's study where he saw that some of these brain phenotypes, so differences in gray and white matter, and how those were mediating this relationship between prenatal cannabis exposure and some of these later child behaviors. I think looking at those differences in the brain, I won't say changes because again, hard to draw causal evidence from this, but I think it would be really interesting to see if we are seeing differences in brain structure and connectivity that might also be potentially mediating some of these differences.
Right, that is so interesting, and really that is one of those ultimate questions that you'll eventually get to. And let me ask you, with regard to methylation, which is what a lot of the data that you presented was about, I suppose that indirectly, or perhaps directly, that is one potential transmission mechanism. If somehow the exposure to cannabis changes methylation patterns, methylation is about the regulation of genes, when they're activated, how much they're activated, how much protein they generate. So that could be one of the most basic mechanisms through which perhaps the fetus's brain is actually changed even before birth. Am I right about that?
Yeah, absolutely. Yeah, we're seeing this, I just showed a couple of the sites, but, they're both showing increased methylation. And so potentially that's revealing, decreased gene expression. And maybe that's coming at like a particular time during fetal development. Maybe it's in a, down regulating genes in a specific pathway, but that's definitely, I think, one avenue through which this might be affecting child behavior.
Right. Let me just conclude by asking you this general question. You're still a young investigator. Where do you see this research leading, and what do you hope to accomplish in the coming years?
Yeah, I mean, again, just hoping to take this project forward to actually looking at some of the behavior that we've looked at in these infants and seeing whether we see associations between these changes in methylation and child behavior. I also think it's great to see studies like HBCD coming online where, we've really invested in collecting these large longitudinal data sets. And, I think it's also important to be looking at both the child and the parent, right? So I think there's going to be a lot of really interesting research that comes out of HBCD, both through genetics as well as potentially, epigenetics and transcriptomics in the future. So yeah, I'm really excited to, I think, hopefully try and get more at what you were talking about earlier, where, you know, we're always looking at associations and correlations, but it would be nice to try and get closer to causation through triangulating through different methods, looking at these longitudinal prospective data sets, and then really trying to get at the mechanisms underlying this, because, you know, we are seeing these increases in prenatal cannabis use, and it's important to know, how is this potentially affecting the child, and through what mechanisms are we seeing that?
I understand. Well, Dr. Johnson, I really appreciate your answering all these questions, but more than that, giving that very, very interesting presentation about the really interesting data that you've collected so far and that you're trying to, you know, make something out of. You really enlightened us as to at least some of the potential in this research. I'd just like to say to the larger audience that we're asking you to please consider making a donation to BBRF today. 100% of every dollar that you donate for research is invested in our research grants, and we can do this because our operating expenses are covered by separate foundation grants. And this means that when you donate $1 for research, that dollar goes directly to the scientists like Dr. Johnson who are doing this research. And so to make a gift, I hope you would visit bbrfoundation.org, bbrfoundation.org, or call 800-829-8289. This webinar has been recorded for those of you who may have been wondering. And if you missed any portion of this presentation or would like to share it, please visit the events and webinars page on the BBRF website. I hope you will join us again on July 8th at 2 p.m. Eastern Time, when Dr. Helen Blair Simpson will present Transforming Treatment Outcomes for People with OCD. We'd like to thank you, and I'm going to conclude by saying what Dr. Bornstein always does at the end of these webinars, which is to remember that together we can dramatically improve the lives of those living with mental illness and enable more people to live full, happy, and productive lives. So thank you very much for joining us today.